So I am Bénédicte Lebret from INRAE, and I will speak about rearing non-castrated male pigs in organic farming and avoiding boar taint in pork. So first, I would like to start with few words about INRAE. So INRAE is a national French institute for agriculture, food and environment. So it's a merge. It was born in 2020. It's a merge between INRAE, which was previously National Institute for Agricultural Research, and IRSTEA, which was a National Institute for Science and Technology for the Environment and Agriculture. And I think during conception and at birth of PPILOW, we were still INRAE, and we moved to INRAE at the beginning of the project. So INRAE, its missions are to conduct research that serves the society, to produce and disseminate knowledge to help solve major societal challenges, and also to put this knowledge to work, to foster innovation, provide expertise, and lend support to public policies. So INRAE includes around 8,000 permanent staff and around 4,000 non-permanent, including PhDs and internal people. And we have arranged in 14 scientific divisions, more or less on disciplinary approaches. And we have around 200 research units and 43 experimental units. So you have a map of France, not very clear. And you have INRAE labs in almost each region, including overseas. And my lab is located here in the northwest, in Brittany, close to Rennes. And for example, the one of Anne and her colleagues is in Pays de la Loire, somewhere there. Okay. So now my talk about first few words of organic farming. You probably all already know, but it's under EU regulation from 2018, the last one, in force since 2022. So it's endorsed by European policy on a farm-to-fork strategy for a fair, healthy and environmentally friendly food system within the European Green Deal. And some studies have shown that organic food is chosen by consumers for health, food quality and ethical motives, including environmental consciousness and animal welfare. So, some words about male pig farming, both in organic and conventional. In France, but also in other countries, there is a ban of surgical castration of male pigs without anesthesia from January 2022. So it means that only surgical castration with anesthesia, local or general. It is local for France and Denmark, and general for other European countries, in some other European countries, and analgesia. So it is allowed only with these two practices, but then it is more complicated, it's time-consuming, and it's also costly. It does not completely avoid pain during and after castration. Some works conducted in our lab have shown that. And the wound can be a source of infection later, and of course it is still a mutilation for the pigs. So why did we castrate pigs? And what are the main advantages or disadvantages to stop castration? So this is a synthesis of scientific knowledge from conventional farms, but actually it's also applicable for organic farming. So first, there is without castration, no more surgical intervention, so it's positive for the farmer in terms of work, both in terms of quantity of work, but also the kind of work, because it's not always very pleasant for the farmer to practice castration. And also it's of course better for animal welfare. You have a better feed conversion ratio because intact males are more efficient. So you have a reduction on feed costs and also then on the environmental impacts due to nitrogen expression. But the drawback is you have a risk of harmful behavior between males, especially mounts and aggressivity. So farm management has to be adapted. A positive fact is anterior males have a higher carcass leanness, so higher lean meat percentage means a higher commercial value for the carcass both in conventional and organic, because farmers are paid on this trait. And another problem is the risk of boar taint, which is an undesirable flavor or odor of meat in some carcasses of intact males. So these carcasses have to be identified and the meat used accordingly to avoid rejection of the products by the consumers. So boar taint, what it is? So boar taint is mainly due to two molecules, androstenone and skatol. Androstenone is synthesized by the testes, so of course it's specific of intact males. And by contrast, skatol is produced in the gut by bacteria in all kinds of sexes, if I can say, in intact male, non-castrated males and females. But in intact males, the skatol content is higher because male hormones prevent the metabolism, the transformation of skatol in the liver. So you have higher contents of skatol in intact males compared to castrated males or females. So the direct elimination of these molecules are saliva for androstenone because it's a pheromone, so it's naturally to attract females. And for skatol is feces. These molecules are stored in fat tissue, both, and they are degraded in liver and eliminated through kidneys in urine. So, what are the problems? The problem is that almost all consumers are sensitive to skatol. I forgot skatol gives an odor of feces, and androstenone an odor of urine. So it's not very positive. So almost all consumers are sensitive to skatol, but some people are not sensitive or little sensitive to androstenone. So it's a high variation between consumers. And the people that do not detect androstenone are called anosmic. They cannot detect that in the meat or in the fat. So products from boar tainted meat have more or less risks to be rejected by consumers, depending on the consumers, but also the products I will show you. And this means that boar taint carcasses have to be identified on the slaughter chain. So how can we use boar taint in carcasses? Actually, it's a bad odor, but it's safe. So it can be used and eaten, of course. So the higher risk are for the fresh products cooked at home, especially when the pork pieces are fat, because these molecules are stored in the fat. So the higher the fat, the higher the risk. And also it's risky for fat products cooked at home and grilled, especially like sausages, and consumed hot, like bacon or sausages. So especially when you cooked with a high temperature, like grilled or fried in the pan, you can have you can smell. In case of tainted carcasses, you can smell these molecules because they are highly volatile. So this requires the screening and sorting of the carcasses on the slaughter line to optimize their use. Other methods are to, when you identify this kind of meat, you can use it for fresh meat or processed products and to adapt the processing, especially to products consumed cold, like pâté or cooked ham. You can also use masking techniques like smoking or adding spices. And you can also dilute this meat with non-tainted meat. For example, when producing sausages, you can dilute the tainted meat in a batch with non-tainted to decrease the risk of consumer to detect this defect. So how can we detect boar taint at slaughterhouse? So in France and other countries, the method used right now is a human nose evaluation. So it has been implemented by Unifor, which is an independent chain actor for classification. So it's an independent evaluation. And with the help of IFIP, the Technical Institute for Pig Production, so they selected and trained panelists, actually people working at slaughterhouse, to detect and to identify boar taint odors, and they are trained to score, and their aptitudes are regularly controlled. And even each morning they are trained just to check if they are, for example, if they have a cough, they could not smell, so they are replaced by someone else. So this is done online in 2 steps. First, they heat the back fat around the neck, and then they smell, and they score from 1 to 5. And I will show you later results, and in that case, the score was from 0 to 2 because it was done by a private company. So the advantages of this method is that it is easy to implement, even if it needs training of people. It's immediate result, then you can have go or no go on each carcass. It has a good ability to detect highly tainted carcasses, and is cheap. The limits are that there is anyway subjectivity despite training of the two operators, and some boar tainted carcasses might not be identified. I mean the carcasses like in between that may not be identified by this method. So there are other methods in laboratory. We can quantify the concentration of androstenone and skatal in the back fat by a technique called HPLC, high performance liquid chromatography, which is a reference method. But it takes time, especially because first we need to extract a molecule from the fat and then to quantify the molecules by this technique. It needs also skilled lab technicians to perform the analysis. And it's not possible to use online because you need a lab environment and not a slaughterhouse environment. So it's not possible to use for the sorting of carcasses in slaughterhouse. And also, androstenone and skatal contents are associated to a risk of boar taint. It does not mean that with this level you are sure, 100% sure, that's no problem, and with this level, 100% sure you will have problems. It's a level of risk. So extensive research has been conducted since years, and even I would even say decades, to develop rapid online methods to identify tainted carcasses on the chain. And in Denmark, they developed one few years ago, and they implemented with a mass spectrometry method with automated sampling and sample treatment. So it's very fast, robust, and it measures both androstenone and skatal, because the best is to have the combination of the two molecules. But it's expensive, it's €1.30 per carcass. So it's difficult to have something fast, efficient, and cheap. So how can we control boar taint? Actually, these two molecules can be controlled by factors, by levels at farming level. For androstenone, the main factors influencing are the genetics, the age and live weight at slaughter, and also, to a lower extent, the hierarchical status of the animals and the environment, especially the duration of lighting. And for skatal, it's much more the environment with the cleanliness of the pen and the level of air renewal and the feeding. Also, the health status influences and a little bit the genetics. And as I said, there is some positive associations between the level of androstenone and the level of skatal. So based on that, we consider that in organic farming, it would be better to avoid surgical castration to guarantee high welfare standards and, of course, to avoid mutilation of the pigs. But we need to develop strategies to prevent undesired behavior of the animals, especially mounting and aggressions, and also avoid these boar taints in meat and pork products. And the aim is to allow ending of surgical castration in good conditions for animals, farmers, and the consumers. So in the PPILOW project, we worked on various strategies that we tested to aiming to reduce or controlling the risk for boar taint of organic intact male pigs due to androstenone, and then we worked on the effects of pig genotype on slaughter weight, which I will show you in the first study that we conducted in our lab. And another part of the work was aimed to reduce the skatal, and so to consider the effects of feeding on animal management, and this was done in the second study conducted by the partner IFIP. So the first study I will show you now. So we worked on the pig genotype, on slaughter weight, on effects on animal welfare indicators, on meat quality. So in that study, we considered two genotypes. So it was, we worked on, health and welfare indicators, boar taint, and carcass and meat quality from intact male pigs in two genotypes, the crossbreed Duroc par large white sow and the Pietrin par large white sow. So why these two breeds? Because we know there is some breed differences in animal behavior, and this could also be the case for intact males in organic farming. Pietrin is used as standard breed for conventional but also organic. It has a low risk for boar taint, but it's like standard meat quality. And Duroc, we know it has a higher meat eating quality and also technological quality, but a higher risk for boar taint, mainly because it's more fat. So we tried to find out how these two genotypes could affect all these traits. And the second step or objective was to predict the boar taint risk at lower slaughter live weight. So the experimental design was as follows. So we did the experiment in INRAE per organic farm, which is certified organic. It's located in the center-west of France. And we had two experimental replicates, each including one group of intact male pigs per genotype. And at the end, we had 47 Duroc crossbreeds and 34 Pietrin crossbreeds. The feeding were the same for all pigs, with growing and finishing organic diets provided ad libitum, and all pigs had access to hay in a rack. And we had two slaughtering sessions per replicate with similar number of pigs from each genotype at each session. So we did some observation on health and welfare indicators during growing at three different steps: at the beginning, at the middle, at the end of the growing fattening period. We also took blood samples during fattening at the end of from 85 kilograms onwards. We recorded gross performance and carcass traits, and we assessed meat quality traits and boar taint components. So this is just to explain you a little bit more the design. So the pigs were in the pen like this. So they had a feeding area on a satellite floor, then a resting area with 1.3 square meter per pig on deep litter, on straw, and then they had full access to a small yard. So it's organic. The minimum regulations for organic farming for pigs in Europe. So this is a scheme of the pen of the barn. So you had a center corridor and each pen on each side, and this is an outdoor picture of the barn. So regarding the results on indicators of health and welfare, we assess the mortality rate and also indicators directly on the pigs. So the highest difference was we found a higher proportion of pigs with high level of skin scratches in the Pietrin compared to Duroc crossbreeds. We also found numerically higher mortality rate on the pigs with skin wounds in the Pietrin, but it was not significant. And regarding tail lesions, we found a trend for a higher proportion of pigs with tail lesions in the Pietrin crossbreed compared to Duroc at the end of the finishing period. And the higher number of scratches found during growing was also found at slaughterhouse, when we found higher average number of skin scratches on the Pietrin crossbreed compared to Duroc. So these results, especially the skin scratches results, suggest lower aggressive behavior or mounting behavior on the Duroc crossbreed compared to Pietrin. And as I said, we found some indicators of degraded health that were higher in Pietrin, even if it's not significant. So this suggests an improvement of welfare indicators in intact males of Duroc compared to Pietrin crossbreeds. And we also found less time of when doing a test of animal-human relationships. We found Duroc had spent less time to approach an unknown observer compared to the Pietrin, which is considered as a positive effect. Regarding gross performance and carcass traits, so we found similar final live weight and gross weight, and also daily feed intake and feed conversion ratio according to the two genotypes. But carcass dressing was lower in Duroc, and then we had a lower whole carcass weight, mainly due to the higher weight of digestive tract in this breed. And also we found a lower lean meat content, which means a lower commercial value of Duroc compared to Pietrin. This is already known, but and also well known in females and castrated males, but of course we found also it in intact males. And this was due to the fact that Duroc has lower fatness of lower muscle thickness and higher fat thickness. We assessed meat quality traits in the loin, which is the longissimus, so the muscle used for chops and for roasts. And we did not find any difference in ultimate pH, which is a very important trait for pork quality, but we found a lower drip loss, a little bit lower lightness, means less light meat, and a little bit higher value for redness, which means a little bit more red meat for the Duroc compared to Pietrin crossbreeds. And we also found higher intramuscular fat content, which is one of the known characteristics of the Duroc, which is important for sensory quality and usually associated to a higher tenderness of meat. And we also found a trend for a lower shear force of cooked meat, which means meats are a little bit more tender because you need less force to cut a specific piece of meat. So this indicates that overall technological and sensory quality of meat would be improved in the Duroc compared to Pietrin crossbreeds. And we also found similar results in the ham, especially regarding the color. Now, regarding the boar taint components in back fat, so we quantified the skeletal and androstenone in the fat of each pig, and we found no differences in average skeletal content between crossbreeds, but we found higher androstenone content in the back fat of Duroc crossbreed compared with Pietrin. And also important, only one carcass was detected as odorant at slaughterhouse using the human nose technique. It was this one, so it was a carcass from Duroc, but which has the highest level of skeletal, which was very high. And considering the limits of perception, I would say rejection by consumers that have been determined or calculated to be 0.15 microgram per gram of liquid fat for skeletal, and 3 micrograms per gram of liquid fat for androstenone, which are the red bars. More carcasses from Duroc compared to Pietrin would be rejected by the consumers. But again, it's a risk. It's not absolute value. And we also, we knew already that there could be a positive association between the content of estradiol in plasma, which is an hormone, and the content of androstenone in the fat. And we assessed plasma estradiol on all pigs, and we again found this correlation. And based on that, we could consider that plasma estradiol could be used to predict fat androstenone and then to predict the risk for boar taint in the meat. So, based on this correlation, we found that around 50 micrograms of plasma estradiol, you can be below or higher the risk of boar taint linked to androstenone. And you can see here that we calculated that with estradiol content below 50 picogram per milliliter, the risk for boar taint due to androstenone is 0% because all pigs were below 3 micrograms of androstenone. But when you are above 50 picogram of estradiol per milliliter of plasma, then the risk for boar taint is around 36%. Based on that, and also considering the blood samples we took on the animals at various steps of their growing finishing period, we calculated the risk for boar taint when we decrease the slaughter weight. And you can see that for the piétrins, which are in orange on this slide, the risk is around 6% at 125 kilograms or above, and is very low below. But for drug, the risk is much higher. It's around 23% above 125 kilograms, and it decreases when you decrease the slaughter weight, but it's still around 8% at less than 85 kilograms at slaughter. So the risk is higher for dogs than piétrins and decreases when you decrease slaughter weight. This is the risk for androstenone, and I said there are two molecules, and we consider that the risk for scatol would be independent of live weight because we know that it depends on all other factors, and it will be low if good conditions of environment are provided, like clean animals with clean bedding and good air renewal. So to conclude on this study, we assessing pig genotypes and comparing Duroc compared to piétrin. Our results suggest an improvement of some welfare indicators for intact males from Duroc compared to piétrin crossbreeds. We found similar growth performance, but lower carcass leanness of the Duroc, meaning lower commercial value. A higher technological quality means a higher ability for processing, and it is very important in pork because more than half of the pork meat is consumed as fresh as processed products, and in France is 75%. In conventional, but also in organic production. And meat quality traits, especially intramuscular fat and shear force, suggest a higher meat tenderness, but unfortunately we could not conduct sensory analysis in this study. And we also found a higher risk for boar taint in the Duroc crossbreeds due to androstenone. And we found that reduction of live weight at slaughter would decrease the risk for androstenone, especially for the Duroc. And overall, it's better to avoid live weight above 125 kilograms. And now we'll present the result from a second study conducted by our colleagues from IFIP. So the aim in this study was to evaluate the effect of additional straw in the pen and incorporation of 10% of alfalfa meal in the pig diet on the health and welfare of intact males and boar taint. So why these two factors? Because we know that cleanliness of the pen contributes to reduce the scatol content in the back fat and the risk for boar taint. And we also know that including crude fiber in the diet can reduce the scatol production in the gut and can also influence some positive animal behaviors.
We have 5 minutes.
Okay, it's almost finished. So in the design, they had two pig groups on the 4 batches for a total of 165 male pigs. So the control group was fed an organic diet, and the test group had organic diet including 10% alfalfa meal during the last month before slaughter and additional straw in the pen during all along the growing finishing period and also a few hours before first departure to slaughterhouse. So the results shortly on health and well-being, they evaluated different traits and they did not find any significant differences between control and test groups on indicators of health and welfare. Regarding carcass traits, they found a similar whole carcass weight and limit percentage of the carcasses in the test on control groups. So the feeding regimen did not affect these traits. And regarding the boar taint component in black fat, they observed a lower content of androstenone and especially of skeletal in the test group compared to the control. And when we consider the threshold I showed you before, they found that the proportion of carcasses with skeletal over the threshold was higher in the control than in the test group, and the same for androstenone. And the score of human nose at slaughterhouse, a score higher than 2 means tainted carcasses. The proportion of carcasses, tainted carcasses was higher in the control than the test group. So additional straw in the pen and the incorporation of 10% of alfalfa meal in pig diet led to lower skeletal and androstenone contents and lower proportion of carcasses detected as tainted by human nose. So the animal diet and management are effective levers to reduce the risk for boar taint, especially the risk due to skeletal. So a few general conclusions on these 2 experiments with non-castrated male pigs in organic farming. So we consider from our results that it's possible to produce non-castrated male pigs in organic farming with satisfactory growth performance and carcass traits. Overall, in our experimental conditions, the health and well-being indicators suggest satisfactory conditions for the animals who had very few low percentage of lameness and all these kind of problems. The tested levels at farm levels, which were genotype, slaughter weight, and animal wealth management and feeding, influenced the risk for boar taint, but had less impact on health and welfare indicators, maybe except for some indicators in the Duroc crossbreeds. The risk for boar taint was quite low in the first study and higher in the, but was higher for Duroc compared to Pietrain. But we also found positive effects of Duroc, so we consider it's a trade-off, and the farmers has to make a choice according to their priorities. And also in the second study, the risk overall was higher, but animal management and diet can reduce efficiently this risk. So altogether, we consider that it is possible to stop castration and rear non-castrated male pigs in organic farming, provided that risk for boar taint and aggressive behavior are managed by genetics and farming practices. Thank you for your attention. And if you have questions, I'm there.
Okay, thanks for the interesting paper. One question about the feeding part of the test. Which strain or which genetics you utilize in the second test? Always Pietrain or other strain?
Yeah, it was Pietrain. So it means that according to it was a different farm. The first one was experimental one, and the second one the commercial one. And according to the other things like environment, with the same genotype, you can have different levels of boar taint. And the proportion of, clean animals was higher in the first than in the second experiment. Overall, means that, again, cleanliness is a very important factor to reduce the risk of boar taint.
Okay, thank you.
Are there other questions?
Do you have any experience in using tannins in diets of pig to reduce the boar taint?
Okay, thank you for the question. No, I don't have any experience on that, and I don't have laboratory data in mind to answer you. There are some experiments trying to reduce the production of skeletal directly by the feeding of animals, but I think the most efficient one is fiber, because it plays on on the fermentation in the gut, and then they decreases the skeletal production in the gut directly. Otherwise, I said that you can also work at the level of processing of meat, as I said at the start, and you can add spices or different kind of herbs in the products to reduce the detection of boar taint.
Feeding alfalfa was related with the level of fever in the diet, so you imagine that the fever can reduce the boar taint. So my question was related with that. For example, tannin or polyphenols can place the same, the same effect.
Yeah. Sorry, we said I understood time. A different one. Okay, tannins. Yes, there are some results showing that tannins can reduce the production of skeletal in the gut and then reduce the risk for boar taint. Yes. I cannot tell you right now the results, but yes, there are some papers published on that. Yeah. So again, combination of different techniques or farming practices can be useful to reduce the maximum the risk.
We have questions from the audience.
Just a short one. I also related to the fiber. So when you say fiber is affected, is that all type of fibers, or is it something particular, maybe in alfalfa, that would not be present in all types of?
It's I'm not. I forgot to say I'm an animal scientist and meat scientist, not a nutritionist. So it's much more the NDF that has to be NDF content that has to be taken into account to reduce the risk. Yes, I know that fiber is a big family, so it's too short to say fibers, but yes, there has some work to explain which kind of fibers are effective to reduce fermentation and the risk for skeletal production.
The last question. Do you have any experience on heavy pigs? Because of course in Italy we produce heavy pigs for the farm ham production. So by the end, I know that the weight of the animal is one part where we have to manage it, but we have a different rule to produce the farm ham. So any suggestion on that?
Thank you. It's a hard question. Yes, I know it's in heavy pigs. It's a big problem, and especially in pigs include pigs used for production of PDO products like Parma ham. But we also worked with the Basque pigs in France some years ago, and the Gascon and so on is the same. So the weight of the animals, but actually the age at slaughter is important, because with animals are slaughtered after puberty, you have of course a high level of androstenone, and could be also a high level of skeletal. So I know it's difficult for this kind of productions or for these producers, because as far as I know in France, in the local breeds, they are still practicing castration, because it's difficult to produce entire males. Also, females are slaughtered after puberty, so it could be a problem in this kind of production systems. So there are still practices castration.
Thank you, Bénédicte.